morning, and thank you, Mark, for the introduction. Is that on? Can you hear me? Okay. So my topic is this emerging therapies. Clearly, I could spend hours talking about this. Each one has its own. You're going to get really a whirlwind of what's out there. And this is unique because, 20 years ago, this talk wasn't even on the radar. We just didn't have this as sort of new therapies coming. So it's just a very challenging time because therapies are coming along. But as a lot of you realize, you may not be eligible for some of these therapies because of the clinical trial situation. So that's what makes it even more challenging as we develop new drugs. So I have a disclosure here that I'm required. I consult for a variety of biotech companies. I serve on advisory boards. I'm currently the principal investigator for a phase 1/2 and a phase 2/3 intrathecal enzyme trial for Hunter syndrome, that I'll talk about at the end. And I'm currently now I have a phase 1 gene editing clinical trial for MPS II that Chet talked about with Sangamo, so I'll briefly talk about that. Clearly, challenges of MPS are very heterogeneous. There's a lot of different types. So what works for one disorder may not work for the next disorder. We clearly have a mechanism to treat cells, as you heard from Dr. Whitley. It doesn't take a lot of enzyme, and that's one of the key factors, and that's why this area of the MPSs is really, the pharmaceutical companies have jumped into it because there are amenable therapies. The question is how to deliver those. It's important to realize that it takes, in terms of to correct the storage of the glycosamic glycans in an individual cell, you may need only 1 or 2% residual activity. So you need a tiny amount of enzyme compared to that normal 100%. And that's what makes these disorders really unique. You're aware of the current therapies in terms of transplantation and IV enzyme replacement therapy. This is just my view of sort of the world in terms of treatment options for the MPS. We know transplant is the treatment for the severe form of MPS I, but there's clearly no therapy for the brain disease for the patients with Sanfilippo A and B and other Sanfilippos, or for the severe form of Hunter that's approved. And so that's really the big unmet need. Clearly enzymes available, but it has limitations in terms of what it can do. So there's clearly a need for more therapies. I'm just going to skip this just to sort of cover some more time. So what are some of the challenges in treatment? We have no effective therapies for some of the disorders. We all appreciate the kids and parents and adults who are on enzyme. IV weekly enzyme is very expensive and also very time consuming. You can basically spend your day going to and from the infusion site if you have that, getting your infusion back home. So it's really a day a week, and so that's really difficult for lots of families. We haven't really treated skeletal disease, and that's an important aspect. As we saw the short stature pictures that Chet showed briefly. The CNS disease has been difficult to treat, except in the MPS I patients undergoing transplantation, and it's not clear why the other forms of MPS, MPS II and Sanfilippo A and B, have not been anywhere near as amenable. It may be time. That is, the MPS I severe patient presents much earlier than the Sanfilippo patients or the Hunter patients. So it may be the timing, but it's probably more than that. And the rationale for why we don't have a stabilization for the other disorders is not clear, but that's where the big unmet medical need is. So what I want to do now is just talk about future IV enzyme replacement clinical trials that just give you a view of what's coming and what's out there. For those families with MPS VII, recombinant IV enzyme is hopefully going to be approved in the relatively near future. Ultragenics, a company founded by Emil Kakas, who really pioneered the ERTs for the MPS, has really very positive data in a small number and has agreement from both the FDA and the European equivalent of the FDA, the EMA, for a path forward. They have submitted their so-called biological license application, that is the application to get approval for the MPS VII drug, to the FDA in May of this year. And so one would hope in 6 months we're going to see a positive result and approval for patients with MPS VII in terms of IV enzyme replacement therapy. As we realize for the MPSs, there's really sometimes only one product, but now for MPS II, and it's been for a number of years, there's a company in Korea called Green Cross, who has a recombinant human ironate sulfatase, hunterase, that's really the equivalent to Elaprase, and so that's not available in the U.S., but it's available in other parts of the world, and that company would like to have that product available in the U.S., and there may be clinical trials at some point being done in the U.S. to try to get approval. As we alluded to, or Chet alluded to, Alexion Pharmaceutical had a product for MPS IIIB. They had some sort of preliminary early results, but clearly that product is no longer listed on their website, and to my knowledge that clinical trial has been closed. And maybe Chet, if he's still in the audience, Chet leave, I believe that trial is closed. So that was another one of these therapies that has been tried and not successful. Armagen, a company that some of you heard last night, has produced is what Chet referred to as modified or synthetic enzymes, but really modified recombinant enzymes that are designed to cross the blood-brain barrier. They started their clinical trials in 2015, and I'll talk more about that in another section of this talk. Another company that's been involved a little later coming is something called Sobi, the Swedish company that's developing. They've modified the carbohydrate portion of the MPS IIIA enzyme to allow it to have longer circulation and therefore hope possibly treat the brain disease. It's not clear to me whether they're going to do IV infusions or whether they'll eventually do intrathecal infusion. That is directly administered, but there's another product available for the MPS IIIA that's coming along. One of the markers of that, they received orphan drug designation by the FDA in July of this year, just very recently, for the treatment of MPS IIIA. So again, another potential company that will be available in terms of in the future studies. Most likely initially in Europe, but it's not clear where they're going to do their clinical trial. So that's just the status in terms of IV enzyme replacement, what's happening in that realm. I just want to talk about now a series of other strategies that's available, that's coming along in terms of treatment for MPS. Some of these may work in the future, they may not all work in the future, we'll just have to see. So this is a list of those. I'm going to get a very rapid sort of run through, and I'll be happy to take hopefully questions before I finish, and you can give me a 10-minute warning mark. I'll appreciate that, but I certainly am around to talk to people at other times. If you see me, stop me, and I'm happy to talk to you. So the first area is this so-called blood-brain barrier penetration or delivery of proteins. We all have the blood-brain barrier, protect really our brain against a lot of noxious stimuli, and so it's important there. Probably the first company involved was Armagen, based on work by Bill Partridge, to move proteins across the blood-brain barrier. What they've done is basically taking an antibody that recognizes the insulin receptor on the blood-brain barrier, attach that antibody, an enzyme, in this case the MPS I or II enzyme, but it could be for 3A and 3B also, which they've done now, and allow that then by IV injection to cross the blood-brain barrier. It also works physically, so it helps in theory. You could get that same product will treat both the physical disease in theory and the brain disease. And they did an experiment using basically monkeys and taking radioactive Elaprase, the commercial product available for MPS II, and they took radioactive, their compound, they call it ATG-182, and it basically injected them IV. And see on the right-hand side, you really see only this sort of very light yellow, that virtually nothing. They showed over here that it gets into the brain. Where in the brain is not clear, but it gets into the brain in terms of, so it does potentially bind to the blood-brain barrier. It doesn't say whether it gets into cells, but it just says it's into the brain. And they may have more data showing exactly where it is. So they clearly have begun clinical trials. There's been 4 adults with MPS II who've been treated with AGT-182 at 1 milligram per kilo IV weekly. And that study has been very difficult to recruit. We heard last night from Armagen that they now have received sort of approval from the FDA to begin a pediatric study, a so-called phase 2-3 study, which Shire has the rights to that study, so that's most likely at some point we'll hear about a pediatric trial for this product using, instead of Elaprase, to use this product as weekly IV infusions. Now, to my knowledge, there's been 3 adult MPS I patients who have used the similar product for MPS I AGT-182 have been treated in the U.S. and certainly the one patient who's been treated the longest, who's here, and you talk to her, and she's experienced some very positive things in terms of that. Because of the difficulty recruiting, Armagen then went recently, not recently, but probably a couple of years ago, went to Brazil and did an open-label escalation study with adults. And then more importantly, they're doing an ongoing infusion of enzyme weekly in pediatric patients. Some of them have Hurler syndrome. It's challenging to know who has who, and they clearly have reported some improvement in cognitive function, and they're very optimistic about that drug. One of the challenges that Armagen has is for a small company is how do you generate the appropriate resources to do clinical trials, which take a lot of effort and a lot of organization. So we'll see what happens in terms of going forward with some of they also have products for MPS 3A and 3B, but just really don't have the resources to push those at the current time. There's certainly other companies that you probably should be aware of, but may not be aware as much, that are in this also field where to modify proteins. So there's a company called Angiochem, which is developing treatment for MPS 1 and MPS 2. They're coupling the enzyme to a small peptide, which thereby, and that binds to something called the LRP1 receptor, another protein on the blood-brain barrier that if you bind to that, it will take it across the brain, similar to what Armagen is doing with the insulin receptor. And so they have a preclinical trial in place. Many of you may not be aware, there's a Japanese company called JCR Pharmaceuticals that actually started a clinical trial in March of this year, a phase 1/2 trial, in which they're using IV administered, again, an antibody which recognizes the transferrin receptor, another receptor on the blood-brain barrier, and they have coupled the MPS 2 drug to that antibody. Similar to the Armagen concept, Armagen is using the insulin receptor, but they're using the transferrin receptor. And again, that trial is actually in progress in Japan. So that's sort of the blood-brain barrier delivery or penetration of proteins. Another concept I want to talk about is substrate reduction therapy. Most of you are aware of what enzyme replacement therapy is. The concept of enzyme replacement therapy is to basically deliver the missing enzyme to the lysosome and have that lysosome then stored material be broken down by the enzyme administered, where in contrast substrate reduction therapy, substrate refers to things that are stored, in this case the idea to decrease the amount of the abnormal material going into the lysosome. This is the abnormal storage of glycosaminoglycans for the MPSs, and thereby, in theory, need less enzyme to help break that down. One of the advantages of substrate reduction therapy compared to large molecules, that small molecules, as you heard from Dr. Whitley, can cross into the blood-brain barrier much more readily than large molecules can. The goal of substrate reduction therapy for the MPSs would be to decrease the amount of glycosaminoglycans that have to be turned over or recycled, and or to look at glycolipids, which also accumulate, particularly in the brain, and probably it's one of those toxic compounds. If you could decrease glycolipid turnover, we could maybe improve the damage or decrease the damage that would occur. These are just some examples of glycosaminoglycans. There's a variety of them. They have all sorts of important biological functions. So it's not like you can get rid of them from the body. So in terms of substrate reduction, one of the challenges is you can reduce somewhat, but you can't totally get rid of them, because otherwise it's not compatible. So it's really, you got to draw that very fine line and find that sweet point where you can some reduction, but not too much that cause damage. Because we have to have GAGs for normal function. It's just the problem with the MPS is that when these compounds need to be turned over, they can't be turned over properly, and they get stored and therefore result in damage. So there is no approved medication in terms of substrate reduction. Genistein, clearly the product that's been reported to inhibit GAG synthesis, and certainly in some cell culture models have some potential benefit. There's an ongoing clinical trial in the UK in terms of this testing genistein in the MPS patients. I was at the UK MPS meeting a few weeks ago and hopefully was going to hear about the results. They're not going to hear results yet, so unfortunately we have to wait a few more months to hear whether genistein in the MPS population has any clinical benefit. It's a double-blind trial, so we won't know until they really sort of finalize the data, and it may be 6 months or longer. There's also a company, Therapies, who's developing a crystalline form of genistein. They're using it for another indication, but in their website they're talking about potentially using genistein in the MPS too. So who knows, that may be another, if the UK genistein study is positive, you may see now other trials with using genistein. One of the challenges is the purification and how to deliver genistein compared to lots of different forms of it. In terms of substrate reduction, BioMarin bought out a company called Zaccaron Pharmaceuticals a number of years ago, and they were developing small molecules to try to do that. What they were attempting to do is to develop a molecule, for example, for Hunter syndrome. In Hunter syndrome, the key storage is a particular type of glycosaminoglycan that has a so-called 2-O-link sulfate. That is just the chemical way for the way the sulfate is hooked to the sugar molecule, and ironate sulfatase removes that sulfate molecule right here, and therefore, and they're trying to develop a molecule which prevents the body from ever making a 2-O-link sulfate. In theory, if they could do that, you still have the glycosaminoglycan there, but you could have basically a molecule which prevents the need from ever having ironate 2-sulfatase. So in theory, it's a fantastic idea. Whether it'll succeed or not, I don't know, but I know they've been working on it very actively for many years, and BioMarin is very good at not telling us what they're doing, which is okay. So that's substrate reduction. So another term that some of you hopefully will start to hear, and I talked last year, is called stop codon read-through. Dr. Whitley alluded to this therapy. So what this is, I'm just going to go to a schematic to show you. So we all make proteins, and if you go back to your basic high school biology, how many remember your high school biology? You learned something about protein production. You didn't realize how important it would be years later, but what normally happens is that we have a messenger RNA that's coded from the DNA. This messenger RNA basically can then make the protein as it reads along the messenger RNA, and you get a full-length functional protein. The normal process is there has to be, this protein only can be so long, so you have to have a stop at some point. And so we normally have a normal stop. What stop codon read-through is small molecules that allowed you to read through a premature stop codon. Start back. So if you have a premature stop codon as a cause of the mutation, what that results in a truncated protein which is totally inactive. And so what happens is now if you have a drug that can read through this premature termination codon, you can get some normal length protein. And so that's the concept beyond stop codon read-through therapy, is that you have a possibility. And this is a drug, in contrast to enzyme therapy, where you have to have the enzyme unique to that particular disorder. This case, stop codon read-through therapy would work for anybody who has a mutation that causes the stop codon. So it's more universal in terms of how it would work. So there's a company called PTC that Chet alluded to that has developed a drug called Adalurin, designed for stop codon read-through. They've done clinical trials in Duchenne muscular dystrophy and very active in cystic fibrosis. They have authorization in Europe, so they have approval in Europe to treat patients with nonsense mutations in Duchenne muscular dystrophy who are 5 years and older. One of the important features for the MPS community, greater than 70% of MPS I patients have one of their two mutations is a stop codon. So in theory, 70% of patients could benefit from a drug like this if it was beneficial for MPS I. The PTC Therapeutics had begun a clinical trial in Europe in 2016 and trying to enroll patients. They have had very strict enrollment criteria, never enrolled anybody in the site in Germany, and I'm not sure what is happening, but looking at their website, they no longer list MPS I as a product in terms of a clinical study where they have other things going on. So it looks like they're no longer interested or no longer have the capability. There's another company, though, Elox, who has filed orphan designation for their product called ELX02 in November of 2016, and I know they're very active, interested in working on this, so I would hope within the next year there'll actually be clinical trials for MPS I using oral medications to help prevent read-through stop codons and give a little bit of residual activity. Currently in the animal models, this type of drugs work really phenomenally well and get significant. So there's another company that we'll see or hear about in the next year or sooner in terms of clinical trials for MPS I initially. You heard a lot about gene therapy from the previous talk. What I'm going to do is just give you some of the highlights of what's out there. So what is the status? So the first one I'm going to talk about is lysogene. You saw some of that data, but lysogene is treated for patients. 1 year follow-up, you heard, showed the patient's tolerant procedure and immune suppression without problems. They're currently scheduled to do a phase 2-3, as we heard last night, worldwide. Hopefully that will start in 2018. They're committed supposedly before the summer of 2018 to start. This is basically using direct brain injection that Dr. Whitley referred to. Another trial in Paris similar to that with MPS IIIB has been performed. There's no published reports, but actually Patty Dixon, who was sitting next to me as we were listening to Dr. Whitley, their paper just came out in terms of the Lancet Neurology, so I can't speak about that, but they supposedly tolerate the injections, since I just heard about that paper just today. In terms of MPS IIIA gene therapy, Abiona, who's also at this meeting in terms of a company, has done IV AAV9 gene therapy and vectors developed by Haiyan Fu and Doug McCarthy. They treated their first patient nationwide in 2016. I believe they dosed 6 patients already. They're going to try to go to other sites in the world. I'm not sure if they're going to do any more US patients. Again, they're using IV-administered AAV9, which is a adeno-associated viral vector that in theory crosses the blood-brain barrier based on data. And they have some early data saying there's some potential biochemical changes. I think it's just too early to see how clinically effective it's going to be. So again, lysogene, this is now sort of future trials. Lysogene is planning this multi-centered trial, both in Europe and the US. Another company, Unicor, had planning an intrathecal trial for the 3B. I'm not aware of that trial is going forward anymore. Samantha, maybe is that trial happening? The 3B? Okay, I'm not aware of it. That company's site no longer lists the 3B trial, and so I don't think it's going forward personally. Another one you may be aware of for Sanfilippo is a company called Orchard Therapeutics. It's planning an autologous, I'll say it and I'll tell you what it is, an autologous hematopoietic stem cell transplant ex vivo gene therapy. So what they're going to do in this case is take patients' bone marrow cells, infect them with a lentivirus vector, not an AV virus, but a lentivirus vector in the laboratory, and give those cells back to the patient, and then see if that actually now dramatically improves the outcome. In this case, the cells in the brain will have a chance to produce much more of the way of the enzyme, because they'll be overexpressing, and that may see a result. So with that type of protocol, this autologous ex vivo gene therapy using transplantation, has worked tremendously well for MLD, metachromatic leukodystrophy, another lysosomal storage disease with severe neurological involvement. So the potential is they're talking about actually a trial starting in 2018 in Manchester. One of the age requirements, you have to be young, potentially under 2 years of age to be in that trial. Abiona, the company doing the MPS IIIA trial, is hopefully going to be starting relatively soon. I don't know how soon. That trial in terms of for MPS IIIB, again, an IV-administered AAV9 gene therapy trial to try to treat the brain disease. In terms of other gene therapy trials for CNX, Regenexx Bio, who's also here, is planning a phase 1/2 intrathecal 9 gene therapy trial where they're going to potentially inject into the base of the neck a one-time dose for both MPS 1 initially and then MPS 2. The question whether these will be adult patients or not is not clear. They haven't filed their the investigational new drug application yet, so we won't know until that is filed and they get approval what those protocols are. I asked the people last night, and they really couldn't really talk about the protocols, so they're not finalized. So most likely that this will be a injection into the base of the brain. Whether they can do pediatric patients or have to do adult patients, that's up to the FDA to make that determination. Another European company with people in Barcelona are developed a MPS gene therapy trials for MPS A, M3A, 3B, and MPS 2, where in this case they're going to do direct injection into the ventricle space within the brain. And so one of the things you may be hearing is, why are all these different types of methods of delivery? IV delivery, direct brain injections, injections into the ventricle fluid, injections into the neck. We really don't know what the optimal means to do this in humans. Animal studies are all very positive, but the question is, what is the best mechanism to get gene expression in patients is still unclear. There's also for Hunter families, there's another MPS 2 phase 1/2 clinical trial using IV AAV9 gene therapies in the planning stages at Nationwide. One of the major challenges in this trial reflects that is how to raise the money to make the vector. Typically, you need to have $1.5 to $2 million to make the vectors in these clinical trials, and that's been one of the limitations for so-called investigator-initiated trials. So in other gene therapy trials, so it's just incredible what's out there. The Italian group, is doing IV AAV9 gene therapy for MPS 6. As I said in the UK, they should have actually dosed their first patient in the last week or two to see what happens for that disorder in terms of, do they get enough enzyme expression? They're not looking at brain disease for the MPS 6 patients. They don't have that, but question, will that actually benefit the physical disease? There's another company who has preclinical studies going on. It's called Immusoft. What they're developing is they're trying to take cells that make so-called plasma cells that really make protein or antibodies, but they try to modify these so they live long term using gene therapy to correct those, and basically as another way to do enzyme treatment for MPS 1. So again, it's incredible the number of companies, and I probably don't have everybody listed here, but this is the best of my knowledge in terms of what's out there in terms of happening. So it's just, what's going to happen in the future. We're going to see a very different meeting where hopefully we'll have a lot more data instead of just saying this is what's possible. You already heard about Sangamo, and I'll briefly talk about they have the technology for MPS 1 and 2. Sangamo now has MPS 1 sites, and my MPS 2 site at UNC is open for the gene therapy trial, as well as the Harmat site for MPS 2 is open for the, I think it just recently got approval. So again, what their design is, as you heard from Dr. Whitley, is to really to use the liver as basically a means to produce enzyme instead of having IV enzyme produced in a factory with $50 million, $100 million factory, to basically use the body to produce the enzyme. They're using the liver, as you heard. And I just want to show you the MPS 2 data. He showed you the MPS 1 data. So if you look at an animal, the MPS 1 mouse model, 21 days after getting gene therapy, you see incredibly high liver enzymes. Here's what's normal. You can see here very elevated plasma enzymes, and the same way with basically complete reduction of glycosaminoglycans in spleen levels, for example, which are hard to reach. So they have a potential, they can make enzyme. It appears to be beneficial in terms of its formulation, and so we'll just have to see what happens long term. Will we get long-term expression? We'll need to do immune suppression to try to prevent the body rejecting it. There's clearly limitations in terms of who's eligible because of antibodies to these AAV that are current in humans, and so we'll just have to go forward and do the initial trial to see how it works. So anti-inflammatory therapies. How are we doing, Mark, for time? Okay, thank you. So anti-inflammatory therapy, again, there's lots of sort of issues related to potentially increased inflammation as a cause of some of their problems. There's a drug called pentosan polyphosphate that's approved as Elmiron. There's been small studies investigated in both Germany and Japan in terms of what's happening. Currently, there's a clinical trial using subcutaneous. One of the problems, Elmiron is not very well absorbed. It's great for the bladder issue that it's approved for, and so they're going to potentially try to use some sub-Q preparations in Germany to see if that really improves benefit. Patty reminded me that there's also an ongoing study recruiting, and she'll talk about it in terms of another anti-inflammatory molecule, and she'll talk briefly about that in her talk. So I just want to end up in terms of intrathecal, where a lot of you know I've spent a lot of time over the last 6 or 7 years involved in intrathecal ERT. I'm going to go through this relatively quickly. Again, the blood-brain barrier is the reason for it being there. Just to remind people that 5 years ago, the term intrathecal was probably a foreign word to this community. Now it's really no longer a foreign word. It's something that, it's the idea of you're putting something directly into the spinal fluid to bypass the blood-brain barrier. You know, the indications for this are really, two central nervous system manifestations and spinal cord compression due to dura thickening. This is a picture I've shown, other people have shown many times, of a very tight cervical spinal cord because the dura becomes very thick and really wraps around, compresses it. It really has some benefit for intrathecal. Intrathecal has some benefit for the dural thickening, but it's limited, but it appears to be safe. One of the challenges that both for MPS 1 and MPS 6, you actually need to dilute the commercial drug with artificial CSF because it's never designed to do it directly. There's been intrathecal ERT used at the University of Minnesota in related to the cord blood transplants, and that study is, I think it's completed, the data is not analyzed, I'm not sure they're enrolling any more patients. Clearly there's a clinical trial of intrathecal enzyme replacement for cognitive decline at Harbor UCLA, and I don't think that's recruiting anymore, is that correct, Patty? So that's no longer recruiting, so I need to change that. There was an intrathecal trial that some of you in the audience were involved in terms of children for MPS 3A sponsored by Shire. Chet alluded to that. That trial has been stopped really to lack of clinical efficacy based on the 1-year data. There's certainly a lot of controversy whether they should have gone longer, but they clearly did stop it, and it sort of raises the threshold for what needs to be done for MPS 3A. As Chet alluded to, BioMarin is doing a clinical trial where they're putting, for MPS 3B, the missing enzyme directly into the spinal fluid or around the brain, and they're using a peptide that's derived from an insulin growth factor that they fuse the MPS 3B enzyme to this peptide to allow better uptake. The trial initially started in Europe. It's given by a drug delivery device that's different from the MPS 2 trial and MPS 3A trials, and it's basically using an intracerebral ventricular injection, that is putting the enzyme directly into the space within the brain, within the ventricle, versus a lumbar puncture is putting into the same fluid, but a lumbar puncture is in the lower back. So it's a lot farther away from the brain. They're using a reservoir called an Ommaya reservoir. Basically what this is a place where you can inject into, and you have put underneath the skin by a neurosurgeon a burr hole in the skull, and then the tubing goes into the ventricle, similar to a ventricular peritoneal shunt. This has really been well tolerated in another group of disorders called CNL2s, that they have now approval for that, and so this has potential to allow this MPS 3B enzyme. BioMarin is starting a natural history study in the US, and some of those patients, depending on their eligibility, will then be eligible to go into the intrathecal trial in the US down the road. So that's another potential treatment for brain disease. So this is just a whole summary of some of those trials I talked about. I won't go through it because of time. What I want to do is briefly talk about the MPS 2 intrathecal trial. Again, it was a trial for patients with severe MPS 2. They had to have cognitive impairment, IQ, effectively an IQ less than 77. We were doing monthly injections. We tried initially 3 different doses. patients had to be continuing on Elaprase, and we randomized 16 patients to study drug or no study drug for first year, and now we're doing long-term. We used a reservoir. This initial one we used, the Smith Medical. We're using something different now. One of the struggles is the reservoir. Patient demographics who we enrolled were basically now, were initially 3 to 11, had a wide range of cognitive abilities, a lot of different mutations. What we saw in terms of the most promising, we got a very rapid reduction of glycosamine glycans 1 month after the first dose. This is just showing you about a year ago the clinical data saying that some patients have done quite well. We estimate this is 12 IQ points lost per year if this is what happened normally, and so some of these patients have done dramatically better. This is actually looking at IQ, or effectively an IQ. My clinical experience, these patients have stabilized, they're not continuing to decline anymore. Some are not getting a lot better, and so the key is really when you start. I'm just going to go in terms of just saying there's a pivotal study that's been fully enrolled, phase 2/3, a worldwide study that was fully enrolled in September. As of the end of this year, we should hear the results of that pivotal study in terms of late November, early December, and that will gear what happens. I know a lot of you asked me, is compassionate use available? And it won't be available unless we see good results from the clinical study. Thank you for your attention.